All right, so hello everybody. Welcome to the latest edition of our journal club. Like I said before, this is a very exciting time because today we have two labs here to present a collaborative paper. So first of all, we have Craig Gorjun, who leads the Cardiovascular Imaging Research Lab at Purdue University. If you want to say hi, Craig.
Everybody, thanks for letting us present some of our work. It's been an awesome collaboration with the Miller Lab at Tulane.
And then we have Jen Anderson, who's a PhD student in the Gorjun lab. And it looks like this is one of multiple different projects that she's been involved in. I don't know if you want to say hi to Jen.
Sure. I'm really excited to be here. I know Cassie is going to be presenting most of our work, but happy to answer questions at the end as they come up.
Yeah, and then we have Kristen Miller, who leads the Biomechanics of Growth and Remodeling Lab at Tulane University. She does really nice work in multiple areas of biomechanics, especially in reproductive biomechanics. If Kristen wants to say hi to everybody.
Hi, everyone. Thanks for the opportunity. We're excited to show our collaboration. Yeah, thanks.
Yeah. And then finally, we have Cassie Conway, who's the first author of the paper and is our lead presenter today. She is a PhD candidate in the Miller Lab, and she got her bachelor's degree from Virginia Tech in 2015. And we just found out that she's about to graduate and move on to a postdoc at UC Irvine, which is really exciting. So I'll have Cassie take it away. And then if people would like to ask questions during the presentation, please speak up. I think Manuel will probably be pretty active in the chat, keeping his soft tissue biomechanics class students in the loop. And then we'll also have plenty of time for questions afterwards. So Cassie, please go ahead.
Alrighty, well, thank you very much for the opportunity, and I'm excited to be presenting your first collaborative paper for this journal club. And I'm excited to talk a little bit more about some reproductive biomechanics of the cervix and uterus. As a little bit of a refresher, I wanted to start off talking about normal female reproductive anatomy, where we're going to majorly highlight the cervix and uterus in both a human and mouse model, where the cervix is going to be our center organ. Cervix is also another term for neck, so similar to cervical vertebrae, it's considered like the neck of the reproductive system or the neck of the uterus in some texts, where it is partially inserted into the lower part of the upper part of the vagina and acts primarily as a barrier between the external environment and the internal uterine environment in the non-pregnant state. And then moving into the uterine body, which you'll notice is slightly different between the human reproductive anatomy on the left and the murine reproductive anatomy on the right, where it's interesting, most animals, most mammals have two uterine horns, as opposed to very few mammals have a singular uterine body. And as I'm sure you know, the uterine body mainly functions outside of pregnancy to build up a highly nutritious endometrial layer that can support a zygote should it implant. So the cervix and the uterus both have unique roles in reproductive function, specifically looking at fertilization, menstruation in mammals that do menstruate, pregnancy, and postpartum. However, historically, the role of the cervix has been debated and relatively understudied, where the cervix has been either one, considered a primarily collagenous stiff organ with relatively little overall function towards the reproductive tract, or two, considered just an extension of the uterus itself without much outside function or structure from the uterus. So it's either been one, considered that it doesn't do much at all, or two, considered it's just an extension of the uterus itself. However, recently, in the last 20 years or so, we've been starting to see more and more studies that the cervix has its own individual microstructure and as well as its own individual function separate from the uterus itself. And so there is really a need to compare and contrast the microstructure and function of these two organs and look at how regional changes throughout the reproductive tract exist. What we do know is that within the cervix and uterus wall, there are 3 primary layers. There is a innermost epithelial, or in the uterus, endometrial layer that is primarily epithelial cells and is a innermost lining amongst the lumen. And then a center layer, which in the cervix is known as the stroma, which is primarily collagen with some smooth muscle cells. Or in the uterus, there is a myometrial layer, which, as the name implies, is primarily smooth muscle cells. And then finally, there is a outermost serosal or adventitial layer that is primarily connective tissue. Then, speaking throughout the layers, the Ecm constituents, extracellular matrix constituents, as well as the cellular constituents such as smooth muscle cells, fibroblasts, or epithelium, are distributed throughout the layers, with the middlemost stromal or myometrial layer containing the most collagen and smooth muscle cells. For the cervix and uterus, we'll be primarily focusing in on the passive extracellular matrix components with collagen 1 and collagen type 3 as a major focus, as well as elastic fibers. And then considering glycosaminoglycans and hyaluronic acid to have a much lower percentage in both of these organs and to not contribute as much to the mechanics of the tissue, where from a biomechanical perspective, collagen resists tensile loading. The elastic fibers provide resilience and recoil to the tissue, and then the glycosaminoglycans and hyaluronic acid will sequester water and resist compression. So what we do know is that the microstructure of soft tissues and the organization of that microstructure dictates the overall geometry and function of a tissue. In the case of the reproductive system, if we have a overproduction or underproduction of certain types of extracellular matrix constituents, we may result in maladaptive remodeling, resulting in different pathologies. For example, overproduction or fibrosis in the uterus could lead to a condition called uterine fibroids, where benign collagenous tumors form in the wall of the uterus, which cause a lot of pain and trouble with fertilization, as well as heavy bleeding in women with uterine fibroids. And then on the opposite end of the spectrum, degradation or atrophy of ECM could result in a condition such as pelvic organ prolapse, where either the connective tissues or the pelvic organs themselves begin to weaken with age, and the pelvic organs begin to fall out of their natural configuration and start extruding into the vaginal wall and outside of the body. So there's a almost homeostatic balance of the amount of Ecm constituents and cells within a tissue that is needed to go through normal physiologic function. However, the microstructure, as well as the function of the reproductive system, remains relatively understudied. So there is a need to explore what the microstructural constituents are further, compare them directly between the cervix and the uterus, as well as look at the mechanical properties of these tissues to better understand normal physiologic function. And then we could begin in the future looking at how overproduction of individual constituents or underproduction can lead to pathologies. So to bridge some of the gaps between the structural knowledge, we can use in vivo imaging, such as ultrasound, to get normal in vivo geometry of the tissue while it's in the body. During dissection or during mechanical testing, the tissues themselves begin to retract after the connective tetherings are cut. So this gives us an idea of how the tissue is oriented in the body. Using ultrasound, we can gather 2D images that can be taken along the length of the reproductive tract sequentially, and then we can reconstruct those images into 3D models shown in the image on the right. Ultrasound imaging is also a very beneficial research tool because it's already being used in the clinic for reproductive imaging and is considered safe and effective for both the non-pregnant as well as pregnant state. Yes.
Cassie, can I ask? Yeah, hey. So right now, the right uterine horn and the left uterine horn, they seem very asymmetric. Is this different in a subject-specific manner, or is this generally true for the murine reproductive system?
I think Jen might be able to help a little bit, but my first guess is that the uterine horns begin to go in and out of plane in the mouse. So as they're collecting the data and reconstructing it, it might start looking like it has little folds within it.
Yeah, that's spot on. It is subject specific, and you know, I think we like to think of the uterine horns as like this textbook image, and the reality of it is it's very different from mouse to mouse and even from person to person, you know. So it might look like it has these folds, but it just depends on the animal. It also depends a lot on, you know, there could be some bowel adjacent to that that's kind of moving things around. So great question, yeah.
Awesome, thank you.
Alrighty, let's see. Moving on from there, if nobody else has any other questions at this point. Alrighty, so moving on from there, we can move from in vivo imaging using ultrasound to look at the macro scale structural information to histology to look at the micro scale structural information, where we can use histological stains to identify specific ECM and cellular components, as well as we're able to use those images to calculate the approximate area fractions for each of these constituents for each organ as well. Combining the information about the in vivo geometry as well as the microstructural data with mechanical properties can provide valuable insight into normal function of the cervix and the uterus. And we can evaluate mechanical properties or determine these properties through the use of mechanical testing. Prior research has used uniaxial tensile testing, where strips or rings of tissue shown in A and B are taken from the cervix and pulled either to failure, where the tissue breaks, or to a set stress or strain in the system. And we can use these strips to independently assess direction-dependent properties. So we can take a circumferential strip and pull on it one way, or an axial strip and pull on it individually, but not together at the same time. For our research, we wanted to explore further into biaxial inflation extension testing, which has been used a lot in vasculature and artery vein mechanics. It allows us to keep the reproductive system in its mostly assumed cylindrical geometry, and we can assess both the circumferential and axial properties simultaneously while we pressurize the tissue and stretch it out. It also provides the benefit that we aren't doing as much disruption cutting to the tissue itself, and the ECM to cell interactions will remain mostly intact. So overall, there's a need to look at the structural properties of the cervix and uterus, look at the microstructure, look at the macro scale geometry, as well as looking at the mechanical properties to better understand the function of these two organs in the non-pregnant state, as well as look at how these organs differ from one another. Therefore, we sought to investigate the biaxial mechanical properties of the cervix and uterus within a physiologic loading environment through biaxial inflation extension testing. And then two, we sought to determine the physiologic geometry through ultrasound imaging and look at the microstructure of the nulliparous murine cervix and uterus. To start off with, we did ultrasound imaging. Well, at Purdue, ultrasound imaging was performed on mice that from the C57 lineage that were 4 to 6 months at estrus, where estrus was determined in B here, where the vaginal opening was the largest compared to the rest of the estrus phase, as well as you'll see proestrus is slightly more red and moist. So the estrus phase is more characterized by this opening and a more pink hue to the vagina. Then the mice were anesthetized, placed on a heating pad stage, and then a 2D B mode ultrasound images were taken along the length of the reproductive system, shown here. And then the images were reconstructed into a 3D model of the reproductive system for each mouse. Then the animals were euthanized via CO2 inhalation. The reproductive systems were dissected out and stored at -20 degrees C and sent to Tulane for mechanical testing. To go a little bit further in depth on the ultrasound imaging, we have been kindly provided an video of the imaging along the length of the reproductive tract, starting at the tendinous arc of the levator ani, which is a major muscle in the pelvis, and in the center, we'll be watching for the reproductive tract, and the scan will continue on to the beginning of the uterine horns. So I'm going to let this play for a couple seconds. Where I think, if I'm not wrong, the bladder is beginning to show up here as the dark large spot, and then up above, we're starting to see a little bit of intestine come into view. Alrighty. So with mechanical testing, the reproductive systems were removed from the mice, and then the cervix and the lower uterine horn was isolated from the reproductive tract itself. The vagina covers part of the external os or external opening of the cervix. So using micro scissors, I removed those basically flaps around the cervix so that only the cervical tissue and lower uterine tissue remained. Because there's the two uterine horns, one uterine horn was chosen randomly to be ligated with silk suture so that while we pressurize the tissue, we will not have any fluid flow through this extra uterine horn at high pressures. And then we cannulated the tissues themselves within our custom biaxial inflation extension device with silk suture again, which here is a schematic of our inflation extension device where we have a small basin that we fill with Hanks balanced saline solution at 37 degrees C. Here is a example of what the tissue might look like on the cannula. We have a pressure transducer near the inlet, as well as a pressure transducer near the outlet from the fluid flow, so that we can calculate mean pressure between the two and have an assumed value of pressure within the lumen of the tissue. We also have a force transducer that measures changes in force on this side here, and a manual axial micrometer that we can use to change the length of the tissue. And then from above, not shown, is a camera system that looks directly down onto the tissue, and we could track at the mid-cervical region as well as the lower uterine region so that we can gather the diameter changes with increasing axial stretch or pressure. At this point, we'll take the unloaded geometry, where we will take the length of the entire complex as well as the diameters at this point. And here is a representative image of what the sample would look like while cannulated onto the device. And you can see one of the little uterine horns sticking out here, and then one cannulated onto the device. Yes, Manuel.
Very annoying. But just for my own education, how long is this sample here approximately?
It's approximately 6 millimeters in length.
Oh, I totally lied to my students. I told them it would probably be around 2 millimeters. But that's really helpful. So it's tiny anyways.
Yes, if it was just the cervix itself, it would be closer to 2 millimeters.
Awesome, thank you.
Yes. So moving on from there, to avoid the effects of hysteresis and make the data more repeatable with multiple cycles, we put the tissue through 5 cycles of pressure inflation and force elongation preconditioning. And then we, moving on from there, determined an estimated physiologic length using an assumption from vasculature that there is a energy conserving length of the tissue, that as you pressurize the tissue at this length, there will be minimal changes in force. So using that assumption shown here, we were able to find in the reproductive system an estimated physiologic length that preserved the force over the range of pressure 0 to 200 millimeters of mercury. And then we moved into our pressure inflation testing, which is where we kept the tissue at a constant axial length, plus or minus 2% the physiologic length, and at the physiologic length, and cycled the pressure from 0 to 200 and back down to 0, again, repeatedly for 3 cycles. And we determined the maximum pressure of 200 millimeters of mercury as a pressure that we gathered from a human study where they did intrauterine pressure measurements in women who were menstruating to see if there were any differences between dysmenorrhea or painful periods versus normal periods. And the maximum pressure was 200 millimeters of mercury. As a fun fact, they've done this during labor, and 500 millimeters of mercury is the estimated contraction pressure. So it's just interesting how even outside of labor, the pressure can get that high. And then moving from our pressure inflation test, we looked at what would happen if we cycled the length but kept pressure constant. So we had a tear pressure of 10, 1/3 max pressure 67, 2/3 max pressure 133, and the max pressure 200 millimeters of mercury, and cycled from our minus 2% to plus 2% length for 3 cycles. Then after the mechanical testing, we removed the tissue from the device and put the tissue in a dish and cut out 0.5 millimeter rings from the mid-cervical region and the lower uterine segment, and took images of these rings, and through a MATLAB code, we were able to calculate out the average thickness throughout the ring itself. And this is just a schematic of how the MATLAB code would take thickness measurements from the innermost part of the ring to the outermost part. Moving on from here, we then took another 0.5 millimeter ring from these areas and paraffin embedded them, cut out 4 micrometer slices for histology slides, and then stained them with Mason's trichrome, Picoseries red, and Hart's elastic stain. And then we did our data analysis where from our ultrasound in vivo measurements, we took the images and calculated out what the diameter and cross-sectional areas would be, as well as the thickness. And then also using ImageJ and GIMP, we were able to calculate out the area fractions of collagen, elastic fibers, and smooth muscle cells from our histology slides. Moving into mechanical property data analysis, we calculated out the stresses and stretches throughout the pressure diameter test to create stress stretch curves. And then we fit bilinear curves to the stress stretch curves, which shown here is a representative sample of a stress stretch curve where stress is on the y-axis and stretch is on the x-axis, where you'll see we have a non-linear portion in the tissues, which is considered the toe region, and then a linear portion that is theorized that there's undulated collagen that is becoming stretched out at this transition point and in full tension here. So because of this nonlinear curve, we fit 2 linear lines at these transition points where we can look at the toe region and take the slope of that for a toe modulus and the slope of the linear or elastic region for an elastic modulus. And we can also look at what the transition stretch and transition stress would be. And then we took a model from cardiovasculature that is a Neo-Hookean 2-fiber family microstructurally motivated constitutive model and applied that to the experimental data that we had, where you'll see that this big equation can be broken down into 2 parts, where this first part will be our Neo-Hookean component, with our C parameter being a part of an assumed isotropic term that represents the ground matrix. And then we will have an anisotropic part at the right-hand side that will be assumed to be our collagen contribution, where with this model, the 2-fiber family comes in that there's 2 diagonal collagen families. We're assuming from the microstructurally motivated part of this that within the tissue, we're going to have diagonally based collagen fibers. And we could take the angle of these fibers across the long axis here. So the main thing to take away is there's an isotropic ground matrix dominated component and an anisotropic collagen dominated component for this. And then, of course, we did statistics where we did paired t-tests to look at differences between the cervix and uterus with the structural data, and then looked at 2-way ANOVA for the bilinear fit parameters, and then paired t-tests again for the material parameters. So what we saw from the ultrasound imaging was that the outer diameter measured at the cervix compared to the uterine horns, and you'll see there's both of the uterine horns in this image here, was significantly greater than the uterus. And we also saw that there was a significantly greater thickness of the cervical wall. We also saw this from our in vitro tests where the diameter was greater at the cervix compared to the uterus, as well as the thickness. And then here is representative samples of our 3 types of stains, where we have our Picoseries red stain for the cervix above and uterus below, where the stain looks at different size collagen fibers. Then we have Mason's trichrome in the center, where it looks at collagen and cytoplasm. And then we have our Hart's elastic stain, which the purple will represent our elastic fibers. What we saw from the Mason's trichrome was there is a significantly greater amount of collagen within the cervix compared to the uterus. And similarly, opposite, we saw that there was a decreased amount of smooth muscle compared to the uterus, which is unsurprising considering the uterus is considered a primarily muscular organ. What was interesting, though, was that the elastic fibers were not significantly different between the non-pregnant cervix and uterus, which I found very interesting because in a couple of slides, I'll show you that the uterus is much more distensible than the cervix, and I had thought that perhaps more uterine or elastic fibers in the uterus may have contributed to that, but it turns out that was not the case. So for our Picoseries red images, the dark red and orange within these images represents larger diameter collagen fibers, and then you can't see it as much in the cervix, but there's a little bit of green and yellow within the uterus that you could pick out from these images represent the smaller diameter collagen fibers. So we took the ratio of large diameter to small diameter collagen fibers, assuming that the larger diameter fibers can withstand greater tensile loads. And the cervix, yes, Emma.
Sorry, I didn't mean to cut you off. I can wait until you finish your sentence, but I saw a question in the chat from Callan. I don't know if you want to speak up and ask the question, or I can just repeat it. The question is, how do you estimate the angles of the fiber families?
Estimate the angles of the fiber families.
Oh, and it looks like Kristen has also jumped in the chat. So maybe the question has already been taken care of. But I don't know if a couple of people said that they were also interested. So maybe Kristen Miller could speak up and read what she wrote in the chat, or I could just read it. But
yeah, sure. I can jump in. We can let Cassie take a quick breather. So ideally, this is something that would be quantified using second harmonic generation imaging and actually informed in the model. So unfortunately, the cervix and uterus are drastically understudied. And so for right now, we just use like the nonlinear regression to simultaneously fit the parameters that are like the lump stiffness, the exponential term, as well as the collagen fiber orientation. So that is the first step, right? An initial study, but that is a limitation. And as I mentioned in the chat, so Kristen Myers at Columbia does have some beautiful work with some of her collaborators that have started to quantify the orientation in the cervix, but there's not any data to our knowledge in the uterus. So this is a plea to our biomechanics community if we need more friends in this space to help with just this fundamental characterization data. So this is why we tried two different ways to try and describe the nonlinear mechanical behavior, but there's this pressing need for more biochemical and microstructural information so that we can have a model that better mimics and replicates that. So yeah.
Kristen, on that note, I'm curious, and I'm putting you on the spot here. Do you think this sort of nonlinear regression, i.e. sort of constitutive identification of the angles, will actually yield accurate results? In other words, do you think these microstructural models really have a microstructural interpretation? Obviously, I know you don't know the answer. I'm just curious what your personal philosophy is, because I always struggle with this a little bit when people interpret the parameters of, say, the Holzapfel model or similar models as actually having meaning. We personally have found that generally speaking, they have pretty poor predictive abilities when it comes to sort of collagen fiber orientations and dispersion angles, et cetera. But that, again, is on a very different tissue. I'm just wondering what your personal philosophy is on that.
Well, my personal philosophy is that they should be interpreted with caution and used to inform it, then to help prioritize the next experimental data and then iteratively refined, right? So what Cassie probably skipped over was numerous studies that we did to try different types of models to see which ones were over parameterized. And like the information on the cervix to date is there's potentially a family of kind of axially aligned and then circumferentially aligned. And then of course there's a gradient. So, we tried different aspects, and this is the one that where the parameters were the, I'm gonna put in quotes, like most determinable, right? And having independent, at least local minima for a first step. But no, I would not walk into a clinician and say like it is X, like definitively, right? But we're just trying to do the first step of fundamentally characterizing the tissues.
Awesome.
I don't know if that answers your question. So yes,
it
careful.
Does.
Yeah, it's important to know the limitations of your model and to, interpret carefully. But at this point, we're also trying to take any information to prioritize, because we don't want to take 50 years to catch up to the cardiovascular use of the models either, right?
Thanks so much. Maybe Cassie, are you ready to take it away again, and I'll go back on mute?
Yeah.
Actually, I will ask one more question on the slide. Sorry.
Wait. Oh yeah, please go ahead, Callan. Yeah.
Yeah, I'm just wondering, like, why did you opt for histology over like an assay in trying to quantify these different proteins?
To start out with, we wanted to be able to look at the overall constituents within that area, and then we can also in the future be able to see like the orientation of the fibers as well. Some of the information that we can get from like a whole tissue analysis, like a Western blot, it can tell me that it has collagen, but it doesn't tell me where in the tissue it has the collagen. And I was kind of interested, as well as like we're seeing here, in that the epithelium was not nearly as like dark for this collagen stain as well as if I go back to my earlier one, we see here, we could see the epithelium along the lines just to kind of get an understanding of what the tissue structure looks like. And it is a limitation that we're only taking a 4 micrometer slice. So we're only looking at this one small section of the tissue and assuming that it's true throughout the entirety of the tissue. The cervix has a greater amount of collagen. So there is a need moving forward to include some of these assays like Western blot for collagen or HPLC for collagen crosslinks, which I think your next speaker, Kyoko Yoshida, will talk about a little bit theoretically if she's doing her cervix crosslink work for the presentation. Did I answer your question?
Yeah, that was great. Thanks.
Alrighty, I think I left off just about to say that there was a greater ratio of large collagen fibers in the cervix compared to the uterus. And moving on from here, if we don't have any more questions about the histology results, just a little bit of a refresher. I know we just talked about the bilinear fit, but wanted to bring this back into your mind after talking about the histology, that we're looking at this nonlinear toe region that transitions into a linear region in our stress stretch curves. So what we saw with the cervix versus the uterus is this transition stretch along the x-axis versus the circumferential stress on the y-axis, that there was a significant increase in this transition circumferential stretch, implying that the uterus is much more distensible than the cervix. And then moving into the toe region and linear region moduli, the cervix had a greater toe region and linear region moduli compared to the uterus as well. So the uterus is more distensible and the cervix less distensible and more stiff. Moving into the next portion where we're looking at anisotropy of the tissue, I have circumferential stress stretch curve on the left-hand side, and then an axial stress versus circumferential stretch on the right-hand side, and saw that for both of the tissues, the circumferential moduli were much greater than the axial moduli, and saw that the stresses increased in the circumferential direction versus the axial direction for both organs. And then moving into our microstructurally motivated model, we have the same stress stretch curve shown from previously, with the addition that the blue is a theoretical, the result of the model with the theoretical curve of the data, where the green is the uterus. And overall, we could see that it was a relatively good fit for the cervix and the uterus, with root mean squared errors less than 0.3. And as Dr. Miller was talking about earlier, we had gone through several iterations of looking at different microstructurally motivated models. A popular one that's being used right now is a 4-fiber family model, which is adapted from this 2-fiber family model, where they have diagonal as well as circumferential and I believe longitudinal fiber families. However, the 4-fiber family model as well as the 3-fiber family model were found to be over-parameterized and over-describe the data simply. So we implemented this 2-fiber family model, and within it, we saw that the all of the parameters were greater in the cervix compared to the uterus. Of interest is our C parameters. C would be our Neo-Hookean component, and then C1 and C2 would be loosely representative of the collagen components within the tissue, and suggest that the cervix, once again, is stiffer than the uterus. And we also had, the model had, using the linear regression, assumed that there was, or calculated out there was average fiber angles of 52.3 degrees for the cervix, and 45.6 degrees for the uterus. And kind of leaning into that, there's the anisotropy within the tissues, but maybe slightly more circumferential leaning for the cervix. Oh, and then I brought this back up and forgot to show it to you guys while we were talking about fiber angles, but just a reminder that it's the fiber angles along this long axis. And then in conclusion, we've presented for the first time the biaxial mechanical properties of the murine cervix and uterus in an inflation extension test configuration. We've compared the in vivo geometry using ultrasound, as well as looked at microstructural information through histology between the two of them. And we found that there is a significantly greater cervix compared to the uterus, which is not unexpected, but it's nice to be able to compare the two directly from the same sample, as well as kind of trying to understand what leads the cervix to be stiffer than the uterus. Is it could be this higher collagen content in the cervix could contribute to these higher moduli, as well as material properties and decreased distensibility. However, it was interesting that the elastic fibers within the cervix and uterus were not different, which may suggest that the glycosaminoglycans and hyaluronic acid play a greater role than we expected. During the estrus cycle, we've learned that glycosaminoglycans and hyaluronic acid sequester more water at the estrus phase compared to other phases of the estrus cycle. And then one thing I wanted to theorize on is the anisotropy of the cervix and uterus may play a role in conditions of preterm birth. For example, cervical insufficiency is where the cervix begins to shorten and dilate in early to mid pregnancy, resulting in early labor. However, the exact mechanism leading to cervical insufficiency is relatively unknown, but maybe this lessened amount of resistance in the axial direction may contribute if there's some sort of maladaptive remodeling over time. And then just to bring some of these ideas in the future, what we would love to do would be to incorporate some of this in vivo imaging and 3D reconstruction throughout pregnancy, as well as do mechanical tests throughout pregnancy for these organs and incorporate all of this information into a 3D finite element model that can we can look at the changes in these parameters and geometry over time with a growth and remodeling model incorporated, so that we could predict progression of reproductive pathologies, or even just predict the progression of normal pregnancy, because that's not something that's been very highly investigated as like a full 3D from start to finish finite element model. And here I've just thrown up a picture of a treatment for cervical insufficiency, which is a cerclage, which basically they stitch the cervix closed to keep the fetus in. So there really is a need to push forward our understanding of reproductive health so that we don't have to rely on such invasive methods that are not don't have a great efficacy. And I would like to acknowledge our funding sources, as well as some of our collaborators who are at Ochsner Baptist, who are our urogynecological surgeons, and all of my lab members who helped out along the way, as well as our collaborators from Purdue. And Hamna here did most of the imaging. She unfortunately couldn't be here because she's doing some great stuff at medical school right now. I really appreciate the work that she did. And are there any more questions?
Yeah. Thanks so much, Cassie. That was an awesome presentation. Do people want to just sort of speak up with questions, or I'm sure there are a few.
I can potentially jump in and answer Nicholas's question. Sorry, I was starting to type it, but so the question was if we looked at strain stiffening effects, since we potentially predict more stress at low strains, so we might be able to account for that with strain hardening behavior. So that's a great question. So sorry, you're going to get sick of us saying that the tissue is understudied. So this was our kind of initial kind of collaboration kickoff. And we did at one point look at potentially taking like a linearized material stiffness at like different strain levels to just kind of fundamentally characterize the tissue. But where we have kind of moved towards now is we now have the capability to use a pressure catheter in an anesthetized mouse to actually measure an estimated like in vivo pressure. And so we've combined that with the estimation of the axial length in vivo, and now just take a linearized material stiffness at that point for kind of our physiologic estimate of the stress and the energy stored within the tissue. So we should, and it's a wonderful idea for us to follow up, but now we've just focused so much on this kind of like estimated physiologic load that we have not gotten to fully characterize just the nonlinear aspects of the material.
Thanks, Kristen.
I have a question as well, if I could chime in.
Yeah, please.
As a fellow experimentalist in biomechanics that does a lot of the same experiments, I have to applaud you in the not only the quantity of work that you guys have done, but the quality. So first of all, I wanted to say that I have a sincere appreciation for it. My question is, you're in your mechanical testing, your choice of reference configuration. So what equates to a stretch of 1 in how do you determine a consistent reference configuration between tests?
So for this set of experimental data, the reference configuration was the first point of the first test. We had gone through an initial unloaded estimation, which was determined visually as where the tissue would be neither in tension or buckled. And then, let's see, I'll bring this back up to my dissection. During the dissection, we had placed dots along the length of the reproductive system of 3 millimeters apart. So the tissue itself in the body was about 6 millimeters and tried to keep that repeatable. So with my first estimation of the physiologic length, I stretched it out to 6 millimeters. That wasn't always 100% accurate. I think partially because I had to cut the pubic symphysis and remove some of the other tetherings before I was able to put these dots along the length. It had already started to retract some. But our initial unloaded geometry estimations were buckled during testing. So I was getting a little bit of data black holes, if that makes any sense. The thickness was greater than the outer diameter. So started as a first point with the first point of the first test. So first outer diameter and length to start us off.
I was curious if it'd be potentially possible to use your ultrasound data for an in vivo reference configuration.
That would be.
I know that's a highly overstatement in making a very complex situation simplified, but using the geometry that you obtain to replicate that and then start your test from there.
That would actually be a really great idea to start off with. Jen, are you guys able to get the length from the bottom part to the head? Or
yeah,
is it
this is an excellent question and some of our more recent work that was not included on this paper. So I did a presentation at SB3C last summer on acquiring cervical length based off these models. Cervical length is what's currently used in people to predict preterm birth. It's a really poor predictor of preterm birth, but it's sort of what we use and why we sort of pursued that cervical length. But it absolutely could be used in the future. It just was something that we hadn't quite gotten to for this paper.
Yeah, that might be something really interesting to do in the future and see how that compares to our estimated physiologic lengths that we figure out during mechanical testing and compare it to what we're seeing in the body. I do worry a little bit about the transducer kind of causing things to move around a little bit, but that still would be interesting.
That's
The
the type of
the
thing
ligation,
where
yeah.
you have to make sure your transducer pressure is consistent and you're not pressing too hard into the animal, because that would certainly impact length as well as width and all of those other metrics.
Yeah. Well, thank you very much. Really enjoyed this presentation.
Thank you for your questions.
I'll add just real quick a comment that this was a really fun study for us because we were able to provide a small amount of in vivo assessment that then you really built on and did a great job characterizing ex vivo. And even if it's just a small amount of data, I think the in vivo characterization with ultrasound, or other people use MRI and other techniques, right, provides at least then an assessment of what's happening in the body and whether you have to build a little bit of a gel bridge so that you're not pressing down, or even, Jen, like we could do some more studies on the supine prone imaging comparison to make sure that body position or how the transducer is positioned affects geometry, whether it's length or thickness or area, right? We can do all that characterization in vivo so that when we're thinking about a true biomechanical ex vivo characterization, you at least have an idea of what's happening inside the animal. And I think, Kristen, I'm really excited about the pressure catheter data, thinking about what's happening from a pressure standpoint, knowing that putting a catheter in right might have an effect too once you start inserting it further in, and just the catheter itself, which it's a limitation on that study as well. But I was thinking maybe do some characterization before and after catheter placement to see how that affects geometry or length as well, to see if that could help validate pressure measurements too. But
yeah, those are great ideas, right? And of course, we also have the anesthesia, right, and how that
Yeah,
affects the.
certainly.
But I think that the point that was brought up about like the in vivo reference configuration is a wonderful idea, and in particular, since now we do have that initial pressure catheterization measurement, we can do some kind of like back calculating to estimate and to do some feasibility studies of seeing if we take that kind of, we'll use guesstimated instead of estimated or calculated kind of in vivo, right? We could start to ask that fundamental question of how that changes, as I'm sure all the biomechanicians are all very well aware of, the reference configuration, how you define unloaded in your mechanical test, and the modeling, but the mechanical test in particular is imperative. And as Cassie mentioned, that was something that we spent a lot of painstaking time of trying to determine experimental cues of a consistent and repeatable. And we do also sometimes have like the ultrasound of the geometry in the testing device too, that we can also
with Carolyn's device.
Right? Start to I mean, we'd love to take yours, but it'd be kind of a haul for
I get it.
from each of those. So I think we're starting to lay the.
Can you share what's initial pressure catheter measurements that you're getting? Are you getting up to that 200 millimeters of mercury human estimate?
No.
Okay.
So initially in the C57 saw so the cervix has phasic contractions. So I have baseline pressure of about 10 millimeters of mercury, and then a maximum pressure, I believe closer to 30 to 40 compared to the 200 we saw in the human. So definitely for keeping smooth muscle cells alive, we had to, for our next study, take the pressure down quite a bit. But it was interesting to see that the cervix could withstand that high of pressure from the human study comparatively.
Yeah. Could you do it in a mouse during labor, during actual delivery in a pregnant animal?
There was a study by Jen Harrington's lab, I believe, that I don't think they did labor, but they did put a intrauterine catheter at like close to day 18 to take measurements. And they had a picture in there where like the catheter was pushed up against the wall between a pup. So I'm not Not sure also about whether or not Ayacook would allow us to put something up in there while they're giving birth. I don't think the mouse would like it either. And anesthesia from that study showed that it slowed down cervical and uterine contractions. So probably couldn't put them under anesthesia either.
Sure.
We're seeing collaboration in real time. So
There you go.
it's exciting. Yeah. Thanks so much for the presentation, Cassie. I don't want to keep anyone after 12. So we might have time for one final question, or we could just end it here. But
I have a question. Hi, Cassie. First of all, great work. And I think all this discussion about the in vivo state is so important because like, I think yours paper is like the first one to actually like do any of that along with the mechanics. So I think this work is like really valuable for that reason. So I wanted to make one comment about the measuring pressures during labor. So I think it's really hard to do because then you have to have some like poor grad student that basically has to wait in a mouse house to like figure out when it's going into labor. But we've done it before. You can like give mice like oxytocin to like make it contract. So we tried it at one point, like first year of grad school, and you can do it. So I think it's definitely possible. And like people have those telemetry, like Sarah England's lab has the telemetry, like pressure catheter devices. So I think with those, I think it is possible. So it'd be really cool to do some of that work. But I did have a question. So my question is about your smooth muscle content in the uterus. So I thought it was actually pretty low, even though the uterus is supposed to be a mostly muscular organ. And then I'm also interested in how that relates to how your toe region is so much longer in the uterus, even though the collagen content is like significantly different, but like to me, like 75 and 70% is like, you know, pretty similar. So can you like comment on that a little bit and what you think is like contributing to that difference in the toe region?
Yeah, so to start off with talking about the smooth muscle content between the cervix and uterus, while doing some literature searches, what I had seen was that in the human, there's a lot more drastic percentage differences in the cervix compared to what we've seen in the mouse. Specifically, they're able to see like a gradient increase from the external os into the internal os, where it was getting closer to like 30-40%. And then the uterus was greater percentage than even that. Oh, hello, Kitty. And then, so I think part of it might be the difference in the animal models, and potentially maybe in the mouse, they develop more smooth muscle during pregnancy, but I'm not sure on that front. And then for the toe region, so part of what I'm wondering about is whether or not there's more crimping of the collagen fibers in the uterus compared to the cervix, leading to this increase in the toe region. And it could also be the combination of crimping as well as the slightly increased amount of smooth muscle cells. I do wonder about the role of the ground matrix with the glycosaminoglycan and hyaluronic acid content here, where I'm unsure if there's a much greater quantity in the uterus compared to the cervix, especially at estrus, and whether or not that might play a role in increasing the toe region here. But I think, long story short, there's some more research that we need to do comparing the two organs directly on that scale. And then another thing I hypothesized was collagen cross-linking, like what you had done in your cervix paper throughout pregnancy, if maybe those cross-links are, there's more immature cross-links in the uterus compared to the cervix at this point.
Cool. Yeah, I think the story is like more studies, please.
Yes, more studies, please. Yeah, I know you're interested in the uterus, so if you wanna.
I am interested in a uterus.
Yeah. All right. It is just past 12. So thank you so much, everybody, for the questions. And thank you to all 4 of our presenters, especially Cassie, for joining us today. That was a really nice talk. Yeah. I'm going to stop recording now, and I think we'll call it a day. Thank you.